Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. We are taking a quick detour from our glycolysis and Warburg metabolism series to take a look at a trend that has been all over social media and YouTube. And that trend is the sugar diet. And some of you who may have been with me for quite a while may be wondering, why would this even need to be covered, knowing what we know about cancer? And I wanted to, number one, address it before it. I get a lot of questions on it. I've already got a couple questions on it. And I want to make sure that not one single person in this audience who is looking for an alternative strategy to overcome their disease looks to this fad as an option. And so therefore, I wanted to take the time to lay out a very strong case for why I believe that this is very likely not the best option for you if you are struggling with cancer. Now, as we'll see when we go through this, there may be some people who probably don't click on a video like this or are part of this audience who may want to try it, and it may not be catastrophic for those people. But for the vast majority of people who are part of this audience, it would likely be catastrophic. So therefore, I want to do the best that I can in dismantling this trend before any of you get, I guess the word would be, suckered into it. So without further ado, let's get into it. So the sugar diet. Today, we're going to talk about what the sugar diet is and basically give as many examples as possible as to why, at least for patients with cancer and probably many other diseases, it would be a pretty bad idea. So if you are scrolling through YouTube and you are seeing videos that look like this, don't be surprised because I'm getting them too. And, you know, it's pretty tempting. You're like, wow, I'm able to eat sugar and look like some of these people in these pictures. Why shouldn't I give this a shot? So the first question is, what is the sugar diet? And the sugar diet effectively is a very high carbohydrate diet, either from whole foods, fruits, and I've seen examples all the way to like gummy bears and like literal table sugar, pouring it into your smoothies and stuff like that. It's generally ultra low fat, and depending on the influencer who you're looking at, it could be ultra low protein or it could be kind of moderate protein. And I find that the people who are not interested in kind of like fitness and bodybuilding, they're generally pushing a more low protein diet. And then there are people who are still interested in kind of like building muscle and things like that. They are kind of pushing more of a moderate protein diet. So where does this originate from? Well, I have been kind of aware of this space for quite some time. So you may have seen some of the like kind of original Netflix documentaries like Forks Over Knives or Food Matters and so on and so forth. And they're going to feature some of these people who are writing these books. One of the ones that got my attention early as a young medical student was this Dr. McDougall, who has sadly passed away. And he wrote a book called The Starch Solution. And, he was featured in a couple of those videos. And then T. Colin Campbell and Caldwell Esselstyn and folks like these. Now, T. Colin Campbell's a PhD. Caldwell Esselstyn's a former surgeon. And then his son, Rip, also had the Engine 2 diet. And I actually personally was a vegan for about 2 years. And there was a period of time when I was even following this diet here, the 80-10-10 diet, which was 80% carbohydrates, 10% fat, 10% protein, and mostly raw fruits, or a raw foodist, and or a fruitarian is the other name for it. And I personally had adverse effects to being on it for longer than about 6 months. But because they do such a good job of convincing you this is the only way to live, you kind of like think that, well, maybe I'm doing something wrong. Maybe I'm not doing it, it correctly. And then, so that's kind of like for general health, like, reversal of diseases. Dr. McDougall was big into like lowering cholesterol and diabetes, kind of like metabolic health. Then the next area where you'll see this at, in particular for cancer, is the Gerson therapy in Gerson Institute. And I also, for a period of time, really wanted to believe that this was a viable therapy. I really did. I mean, I had the Norwalk juicer. I bought it. It was like a very expensive 70-pound like steel juicer that they recommend for the Gerson therapy. If you ask my roommate from medical school, he would tell you that I had that juicer and that I was making these giant, quantities of juice every day. If you're what it says here is that you're juicing nearly 15 pounds of produce a day if you're doing the Gerson therapy. I don't know how many people can actually afford to juice 15 pounds of produce a day. Now, to be fair, it is mostly carrots. It's like 13 juices a day or something like that. Yes, see, they consume multiple 8-ounce juices, mainly carrot, apple, and this Gerson green juice. They also do like soups and stuff like that. But it is generally, first of all, it's vegan, and it also is a very high carbohydrate diet. And I will, like I said, I wanted to believe that this could be a viable option for cancer when I was a younger, kind of more naive, didn't understand cancer biology like I do now. And I have now, since being in the position that I'm in now, where I see people who come for consultation, have seen people who've been on this therapy, who've been to Mexico. I think there are a clinic in Tijuana who've paid, thousands of dollars for a week or two to go down there. Um, Nothing bad about the people down there. They're probably wonderful people. I'm certain they want to help people, but I have not seen anybody personally get better by using that therapy, and it makes total sense why you wouldn't get better with that therapy. I've even worse than that had someone come to me in consult who had been on. I just picked this diet because it's the only quote unquote diet that I could find that had a book that was like this. But basically, like the doctor or practitioner or whoever they hired for their advice basically told them for a period of time for like 6 weeks or 8 weeks to eat grapes. Like they only ate grapes. And then once they got off their grape diet, they went to basically a raw food, kind of like a fruitarian diet. And again, it doesn't make sense that it would help at least from a macronutrient standpoint, like from the proteins, carbohydrates, and fats ratios. Now, of course, I'm not knocking, grapes and grape seeds and things like that. There could be, high benefits with those, like because of the resveratrol and other chemicals that are in those foods. So it's not that they're throwing out the baby with the bathwater. I'm just saying that, being on a grape diet, knowing what we know about cancer metabolism, which we're going to go over in detail during this video, does not make a ton of sense. So, what are we going to be discussing when talking about this sugar diet and doing my best at kind of trying to dismantle and derail these diets, at least for cancer? Okay. Like I don't want to speak to them as a whole. I generally believe that for a lot of conditions, a sugar diet, I mean, again, some of these diets are advocating for like gummy bears and table sugar. That is probably universally a horrific idea. Whole fruits, kind of like fruitarian type diets. I think there could be an argument to be made for a certain person living in a certain part of the world. This is where, like, I have to channel my kind of inner Dr. Cruz and talk about, well, if you live closer to the equator where there's UV out all year round, you probably could do a higher carbohydrate diet. Does that mean vegan? Probably not. I think that at this point, after being a vegan and having it kind of damage my health and seeing it damage the health of other people, I don't think I would recommend a vegan diet at this point. But could it be a higher carbohydrate diet for the right person? Possibly. And that's like a discussion for another day. What I want to discuss is people who are dealing with cancer. Okay. That's what this kind of channel has morphed into. And so my point is that I want to head that off of the past before this fad gets any bigger. And you kind of start scratching your head like, could this be for me? Like, I want to make sure that we are certain that it does not. So normal metabolism versus cancer metabolism is the first order of business. Then we're going to hit some of the hypoxia factors, metabolic reprogramming, hypoxia, pseudohypoxia, cancer-related signaling pathways, hyperglycemia, and cancer outcomes. This is critical. Like if you have, higher glucose, do you do worse with cancer or do you do better with cancer? We're gonna look at that. I'm gonna quickly go over aerobic glycolysis because we're in the middle of a glycolysis series, so I'm hitting it even from a more high level or deeper level on those videos. So there's really not a whole lot I want to go into here, but I do want to kind of like cover it. And then, of course, I'm going to give you my final verdict. So first things first, let's get oriented here. So this outer boundary here is the outside of our cell, okay? And inside here is a giant mitochondria. Now, this is not to scale by all means. Inside of a cell, you may have hundreds, if not thousands, of mitochondria, and they would not be this big relative scale. But for our purposes, this is, I believe, from a C-free paper. It makes a lot of sense to look at it like this, okay? So normal cells, this is a non-tumor oxidative cell. So basically, when we have healthy mitochondria and we are not in a disease state, we are what we call metabolically flexible. So we can use glucose for energy, we can use fatty acids and ketones for energy, and we can use protein and amino acids for energy. And that is a process called glycolysis. That's a process called beta oxidation. And amino acids go through either ketogenic amino acids or glucogenic amino acids. And basically, they all end up in the TCA cycle inside the mitochondria, and then they ultimately end up as the substrates necessary for the electron transport chain, or OXPHOS, or oxidative phosphorylation, and that is called aerobic conditions, and that is like the vast majority of our energy system. Okay, this is how we make energy, and we can look at glycolysis and what you're able to get, and there's all these different steps, right? Like we're kind of going over the channel. And in the channel in particular, we talked about hexokinase, we talked about phosphofructokinase, we talked about that other PFKFB enzyme, and we're just going through pyruvate kinase. Well, of course, we'll hit lactate dehydrogenase and some other things as well as part of cancer biology, but this is like glycolysis as a whole. Let me make this a little bit smaller. So, Warburg metabolism, or kind of the cancer metabolism, is different. Okay. Cancer metabolism is not metabolically flexible. Cancer metabolism can only use basically two substrates for energy production. They can use glucose and fructose because it's going to cycle through glycolysis, and it can use glutamine. Okay, those are the two substrates. And why is it like that? Because we have mitochondrial defects. Okay, the mitochondria, which we're able to make the vast majority of our energy when we have normal metabolism and we have that metabolic flexibility, they're not able to do that. Okay, there is a high degree of mitochondrial heteroplasmy, and the electron transport chain is not able to make enough energy to sustain itself. So what happens is, as a survival mechanism, I believe cancer is a survival mechanism gone awry. It's going to rely on glucose and it's going to rely on glutamine. Okay. The issue with that is that it sets the cell up for basically a lack of control, because the mitochondria are heavily responsible for cell cycle, for cell division, as well as apoptosis. So when you bypass the mitochondria, you are bypassing the control mechanisms. And so these cancer cells can grow out of control. The other benefit to aerobic glycolysis is it's much faster. You can make energy much faster using glycolysis and fermentation. The issue is that for most normal cells, if we were to go sprint or do an HIT session on a fan bike or something like that, we have to stop because we can't maintain that. The difference is, and ultimately the lactate that's created for, that anaerobic metabolism can be cleared and you go back to normal oxidative phosphorylation and oxidative metabolism. However, for cancer, we don't do that. So cancer relies on glucose. Okay. So you can already understand probably at this point why being on an all sugar diet would not be a fantastic idea, but we're going to go further. And just looking at a couple other examples here. So we can see here that under normal conditions, when we're metabolically flexible, we have normal mitochondrial structure and function. we're able to be metabolically flexible, and we're using oxidative phosphorylation as the majority of our energy. However, as we get metabolically more unhealthy and we build up mitochondrial heteroplasmy and the mitochondrial structure and function start to deteriorate, the OXPHOS capacity or the bioenergetic decline starts to become problematic for us. Ultimately, we hit a threshold where, or a transformation where these cancer cells start to become reliant on substrate level phosphorylation or aerobic glycolysis or the Warburg effect. And this is just a blown up giant version of that. And this is like another beautiful version, graphic from one of Seyfried's more recent papers where you have a non-tumoral, again, bioenergetics and a cancer bioenergetics, which basically is showing exactly what we just talked about in these last couple of slides. So again, reiterating, a normal cell, if it's using glucose for fuel, will bring in about, let's say, 1x amount of glucose. However, a cancer cell, because of that Warburg metabolism, because of those broken mitochondria, because of the lack of metabolic flexibility, because of that, the cancer cell is bringing in 10 to 30x more glucose than a normal cell, and it's making a bunch more lactate than a normal cell would, and that is also problematic, which we'll talk about. And it's not getting into the mitochondria, and that is basically showing its reliance on glucose. All right. So again, I think that I'm making the point hopefully pretty clearly that at least for cancer, okay, being on an all glucose diet would not be a good idea. That would be like the absolute probably worst thing you could do for yourself. Now it gets worse because hypoxia inducible factors are critically responsible for setting up this Warburg metabolism and the metabolic reprogramming. And so there's a whole host of things that set up the Warburg effect, which we will talk about. But one of the things that is critically important for that is this HIF, or hypoxia inducible factor. And it's responsible for setting up the Warburg effect. It's also responsible for chemo resistance, radio resistance, immune resistance, etc. And the reason is because when the Warburg effect is in effect, we are creating this acidic tumor microenvironment. And that acidic tumor microenvironment is what leads to chemo resistance. It's what leads to immune resistance and immunotherapy resistance. So that's a big problem. Well, the issue is, as the name implies, HIF, hypoxia inducible factors, are triggered or activated by hypoxia. That's the way it should be activated. However, there are other things that can activate them, and that's a process called pseudohypoxia, which we'll talk about. Hypoxia inducible factor, as you can see here, is responsible for cancer cell metabolism, angiogenesis, or the creation of new blood vessels, invasion to other tissues, cancer stem cells, migration, tumor acidosis, tumor inflammation, etc., which all leads to tumor progression. And as it says here, HIF-1 alpha is at the crossroads of hypoxia, inflammation, and cancer. And you see here for those various reasons. Multiple papers, multiple graphics showing very similar things. Now, the difference between this paper and what the other papers are showing is it kind of gives a little bit of a view of what is actually happening here. Okay, so I'm going to move this around here. I'm going to actually see if I can blow this up a little bit so we can see it. So what's happening is that under normoxic conditions, hypoxia inducible factor is degraded continuously. So it's being made continuously, but it's being degraded continuously when there's no hypoxia or pseudohypoxic conditions. However, if there's hypoxia, then what happens is that these kind of chaperone proteins that lead to its degradation get oxidized, and it allows for HIF-1 to be activated and translocated to the nucleus to then, I'm going to make it a little bit smaller because now you can't see what it's doing. Then once it's activated, it starts to do all these things. We talked about angiogenesis, glycolysis, invasion, metastases, apoptosis, and autophagy changes, as well as immune evasion. So it's a big problem. Well, zooming into what it does for us from a metabolic perspective, you see a little lightning bolt here, a black lightning bolt and a red lightning bolt. And the red lightning bolt is where HIF2 is acting, and it's mostly on the glutamine system. So you see glutamine uptake channels, glutamine utilization, enzymes such as glutaminase, glutamine dehydrogenase, as well as it's also involved in the upregulation of some of these enzymes that are responsible for the fermentation of glutamine to succinic acid. And then the black lightning bolt you'll see here is upregulating glucose transporters, all of glycolysis, lactate dehydrogenase, and lactate transporters to really truly set up the Warburg effect. Yet another couple of things that makes HIF1 stabilization or overstabilization dangerous is the fact that it actually increases the amount of mitophagy. So here it says that it works with about, HIF1 has relationships with autophagy. And what it does is it actually activates autophagy, and it will actually create excess amounts of mitophagy or mitochondrial specific autophagy, which leads to a kind of colony destruction of mitochondria. So therefore, you have to rely on glucose even more at that point because there is no mitochondria oxidative phosphorylation to actually happen, which is dangerous. So, what is hypoxia and pseudohypoxia? Well, again, pseudohypoxia and hypoxia are activating these hypoxia factors, which are having a major effect within cancer. Well, pseudohypoxia would be events or circumstances within the cell that are not hypoxia, but mimic the effects of hypoxia. And it has to do with these proteins PHD and FIH1. And what happens is that effectively the actions of these PHD and FIH1 are inhibited by these pseudohypoxic factors, and that allows for hypoxia-inducible factor to be free and not degraded like it's supposed to, and it's going to be activating its gene expression in the nucleus. So we see here that there's hypoxia, that's low oxygen status, okay? But then there's a bunch of other pseudohypoxic conditions. Some of those would be like reactive oxygen species, certain growth factors, oncometabolites, etc. And there's actually a bunch of causes of pseudohypoxia. So one of the ways that pseudohypoxia can be established is through reactive oxygen species, and that having a crossover with NF-kappa B, which is part of the inflammatory cascade. So inflammation can cause pseudohypoxia. Reactive oxygen species or oxidative stress can cause pseudohypoxia. So pseudohypoxia is one of the activators of hypoxia-inducible factor, or I should say, reactive oxygen species and inflammation are one of the ways that hypoxia-inducible factors are stabilized and activated. And looking further, we see that when we have obesity and obesity-related conditions, hyperglycemia, that can lead to oxidative stress, and as we'll see a little bit later, it can lead to inflammation as well. And we can see that there is direct links between hyperglycemia and the inflammation seen in cancer, as well as the oxidative stress seen that kind of sets up cancer as a condition to begin with, and then can drive it as well. And what we also can see is that hyperglycemia will aggravate the microenvironment and promote metastatic ability of pancreatic cancer and probably all cancers, because hyperglycemia can by itself cause stabilization and upregulation of hypoxia factors by itself. And it says here, our results demonstrate the association between hyperglycemia and poor prognosis and can be attributed to microenvironment hypoxia and pancreatic cancer. I think that's a little bit, in my humble opinion, I personally believe that's a little bit short-sighted. I think there's multiple mechanisms of how hyperglycemia can worsen your prognosis, and I'm going to go over that. But hypoxia factor is a major problem. And we see here that when there is euglycemia or normal glycemic conditions, there is 1x amount of protein expression of hypoxia factors. And when you have hyperglycemia, and it's not defined what hyperglycemia would be in this case, is it like severe hyperglycemia? Is it mild? But the bottom line is hyperglycemia is known to upregulate the hypoxia factors. So a big problem. There's also other evidence suggests that hyperglycemia, inflammation, and diabetes is a major cause of pseudohypoxia. And they have nice little graphics about how hyperglycemia can upset the NADH ratios and create a redox imbalance, a pseudohypoxia, and ultimately oxidative stress, which leads to HIF stabilization, and in this case, cell death and tissue destruction, because they're talking about kind of a diabetic nephropathy, I believe. But my point is that there's hyperglycemia can activate HIF by itself, and HIF, when it's stabilized, can wreak havoc on our chances of developing cancer, but also in setting it up and setting up the Warburg effect, but also in making it worse. You can see here oxidative stress is related to inflammation, and oxidative stress at low levels will cause an antioxidant response. At higher levels will lead to inflammation. And there becomes this kind of like vicious cycle between oxidative stress, activation of inflammatory kind of gene products, inflammatory cytokines. Those inflammatory cytokines can then increase reactive oxygen species, and then there becomes a vicious cycle between oxidative stress and inflammation. And I think that most people know that chronic inflammation is pretty damaging, and it can lead to most diseases or contribute to most diseases, and it can contribute to all stages of cancer. So definitely we don't want to have, we don't want to give cancer any solace. We don't want to give cancer any fuel to the fire. And definitely hyperglycemic oxidative stress, hyperglycemic inflammation is going to be adding fuel to the fire, and we don't want to do that. So there is kind of well-known relationships between hyperglycemia and inflammation. It says inflammation is a form of body's immune response against certain pathogens, toxic particles, and harmful stimuli. It is marked by swelling, heat, pain, redness, loss of function, and increase in these pro-inflammatory cytokines, chemokines, and immune cell recruitment is found in hyperglycemic conditions, which promotes inflammation. Also, high glucose activates endothelial cells, generates oxidative stress, deregulates or dysregulates nitric oxide, and advanced glycation end products, age formation. A study indicates that hyperglycemia has regulatory effects on IL-6 and LDH expression and plays a synergistic role in inducing inflammatory and hyperimmune responses in COVID-19 patients. Alzheimer's disease is a neurodegenerative disorder characterized by loss of brain function. A high blood glucose milieu compromises the blood-brain barrier integrity, as well as increases the promotion of inflammatory molecules like TNF-alpha and NMP9 that promote neuroinflammation. Effects of hyperglycemia: high glucose activates endothelial cells. Basically, this is the caption to this graphic, this beautiful graphic here, where hyperglycemia leads through multiple mechanisms. Here's HIF-1 alpha right here to inflammation. And this is what I would consider also a vicious cycle, because when you have hyperglycemia from being on a sugar diet, and then you have HIF-1 stabilization, that leads to inflammation. Inflammation can then feed back on HIF. HIF is responsible for upregulating all of the Warburg effects. So you can see how there is, a lot of this fits together, right? Now, not to mention, hyperglycemia can also deplete you of certain key nutrients and minerals. So, for example, magnesium, vitamin D, chromium, several of the B vitamins, other vitamins such as iron, calcium, vitamin A have been linked to impaired glucose metabolism increase. And so my point is that other causes of pseudohypoxia. So you see here, you see succinic acid, you see fumaric acid or fumarate, succinate, 2-HG, lactate. These are all oncometabolites that actually will stabilize HIF and lead to a feed-forward mechanism. So, for example, we looked at how HIF-1 alpha increases Warburg metabolism. Warburg metabolism creates a bunch of lactate, and you can see here, based off of this graphic here, that lactate will actually stabilize HIF. So it becomes truly a variety of vicious cycles, really. It's not just one vicious cycle. The reason I bring the succinate and the fumarate up in particular is because when we have deficiencies that are caused by hyperglycemia, magnesium deficiency, B vitamin deficiencies, iron deficiencies, etc., that can lead to these enzymes that are responsible for the TCA cycle not to function adequately, which leads to buildup of fumarate and succinate and other oncometabolites, which leads to hypoxia factor stabilization and ultimately pseudohypoxia, which leads to Warburg metabolism setting up shop in your body. Basically, there's other oncometabolites that can also, like citrate, connotate, alpha ketoglutarate. We already talked about succinate, but many of these are maybe they're not affecting hypoxia factor directly, but they are still oncometabolites. So when you have deficiencies and you have inefficiencies of the TCA cycle due to nutrient deficiencies or other, even toxicities or other reasons, that can also set up shop, cancer as a whole, whether it be stem cell function, immune modulation, etc. So, and again, here's succinate, fumarate, HG, which is a metabolite of alpha ketoglutarate, acetyl-CoA. They can change gene expression as well. That can set up shop for cancer. So the bottom line is that hyperglycemia, as we can see here, it can be a major cause of inflammation, of oxidative stress, of pseudohypoxia, and nutrient deficiencies, which can cause further pseudohypoxia and set up for the vicious cycle to be set up in your body. So again, if you didn't have cancer and you were generally healthy and you were eating whole fruits and vegetables that were maybe starchy vegetables, would it be catastrophic for you? Probably not catastrophic. Could you see how a high carbohydrate diet for long periods of time may not be a genius idea or a good idea? Probably. Does that mean that every person in the world should be on a ketogenic diet? No, I don't believe that. I think it could be more moderate than that. But definitely for cancer, the ketogenic diet makes the most sense for at least for this very good reason. Let's go into some of the cancer related signaling cascades and metabolic reprogramming. So that is looking at, just to be clear, we're going to be looking at some of these key signaling cascades. Now we've been looking at HIF1 alpha here, and HIF1 alpha is independently very important. But some of the other signaling cascades that we've talked about in the past is like the beta-catenin WNT pathways, the YAP or the yes-associated protein pathways, CMYC, the PI3K, AKT, mTOR pathways. Here's the inflammatory pathways, MAPK, as well as growth factors. And I think that's critically important to look at because those are also obviously setting up the Warburg metabolism and the glucose metabolism and aerobic glycolysis. So I think that we should take a look at those and see what hyperglycemia does. So we see that CMYC is a key regulator of the Warburg metabolism as well, and it's upregulated by a high glucose uptake. So you see high glucose activates the beta-catenin and the WNT pathway, which then has relationship to upregulating the CMYC, and then CMYC has its detrimental effects on setting up the Warburg metabolism. What about the yes-associated protein? High glucose can also activate the yes-associated protein, which then can also set up Warburg metabolism. How about this MAPK pathway? Well, high glucose can also signal to upregulate this MAPK pathway. So now we're looking at the beta-catenin WNT CMYC pathway. We're looking at YAP CMYC pathway. We're looking at the PI3K AKT mTOR pathway, as well as HIF1 alpha. We've seen how it activates inflammation, and we've seen how it activates the MAPK pathway. So basically, what we've seen so far is that hyperglycemia by itself can activate each one of these pathways that can upregulate glucose metabolism and lead to Warburg shifts within normal cells and cancer cells. Not good. What else can happen? Well, high glucose can also activate these growth signaling pathways as well. When you have high glucose, you have the insulin receptor. There's insulin like growth factor 1. There's epithelial growth factors. It's related to HER2. Many growth signaling pathways as well can be activated and enhanced by high glucose. What about hyperglycemia and cancer-related outcomes? Well, this paper is titled Hyperglycemia, a Neglected Factor During Cancer Progression. It says here that hyperglycemia, the most important feature of diabetes, may be responsible for excess glucose supply for the glucose-hungry cells, and it contributes to apoptosis resistance, oncogenesis, and tumor cell resistance to chemotherapy. Considering association between diabetes and malignancies, the effects of hyperglycemia on cancer progression in cancer patients with abnormal blood sugar should not be neglected. Glucose through a variety of mechanisms. Here's the epidermal growth factors. Here's other growth factors. There's the insulin signaling growth factors. It's going to lead to progression of your cancer. Here's the MAPK pathway we talked about as being a key signaling pathway to upregulate glucose metabolism and the Warburg effect. So not good. keep going. The effects of hyperglycemia on the progression of tumor diseases. It says here, epidemiological studies have shown that hyperglycemia raises the prevalence and mortality of certain malignancies like breast, liver, bladder, pancreatic, colorectal, endometrial cancers. Hyperglycemia can promote the proliferation, invasion, and migration, induce the apoptosis resistance, enhance chemotherapy resistance, and tumor cells. Not good. Hyperglycemia and cancer. Hyperglycemia, hyperinsulinemia, and insulin resistance influence proliferation, migration, invasion, and angiogenesis, and anti-apoptosis response in cancer cells, rendering them resistant to chemotherapeutic treatment. In general, hyperglycemic patients become more susceptible to different cancers, including pancreatic, stomach or gastric, endometrial, breast, and cervical cancers. It is evident that type 2 diabetes significantly contributes to thyroid cancer, and research discovered that adding metformin to anti-cancer drug enhanced the efficacy of treating diabetic-induced cancer. In another study showing a carcinogen-fed hamster model combined with high glucose diet showed an acceleration of the carcinogenesis process. Figure 1 is explained here. Basically, we have hyperglycemia leading to the upregulation of growth factors, of all of these signaling pathways, ultimately leading to cancer progression. Okay. Hyperglycemia is not something you want to have if you have cancer. I don't think you want to have it anytime, to be honest with you, because it increases your risk for all kinds of other things. Obviously, diabetes, there is cardiovascular diseases, neurodegenerative diseases, et cetera, et cetera, et cetera, diseases that no one wants to have. But when, especially with cancer, it will lead to cancer progression. It is like putting gasoline on a roaring fire. It is not a good idea. Hypoglycemia and cancer. A study found that hypoglycemia induces apoptosis and reduces cancer cell proliferation through the activation of p53 and caspase-3. A previous study from our laboratory demonstrated that hypoglycemia drug pioglitazone can inhibit breast cancer cell growth in PPAR gamma independent mechanism. Hypoglycemia leads to cessation of cancer cell differentiation, inhibition of metastases, angiogenesis, and colony forming ability in metastatic breast cancer model in vitro. CYP2A, which helps cancer cell proliferation by activating c-Myc, is inhibited by metformin. A study found that the combination of metformin and hypoglycemia activates PP2A, which works through a GSK3 beta MCL1 pathway and inhibits tumor growth. Another study using gastric cancer cells reported that berberine low glucose combination can suppress cell growth through a PP2A GSK3 beta MCL1 signaling pathway as well. Are we getting the picture here? Low glucose does the exact opposite. Low glucose hypoglycemia induces apoptosis or programmed cell death. It reduces cancer cell proliferation through a variety of mechanisms. Okay. So hyperglycemia grows cancer, hypoglycemia inhibits it. Okay. Does that mean that are you going to be saved completely on a ketogenic diet? No. You still need the three-legged stool is still in effect, but it puts you in a much better position to have a successful outcome, whether you're doing conventional therapies or not. As you can see here, hyperglycemia induces chemotherapy and immunotherapy resistance. Okay. Let's keep going. Compared to men who were classified as normal glycemic, those who were classified as hyperglycemic had 2.5 times the risk of lethal prostate cancer and 3.2 times the risk of fatal prostate cancer, aka 250% more likely to develop a lethal prostate cancer and 320% more likely to lead to a fatal prostate cancer just by having hyperglycemia. Keep going. The impact of diabetes on head and neck cancer. Elevated blood glucose levels before or around the time of diagnosis have been reported with reduced survival rates of head and neck squamous cell cancer. Hyperglycemia predicts adverse prognosis in patients with advanced pancreatic cancer. Patients with hyperglycemia had a worse prognosis than those with normal glycemia, 7.5 months versus 8.8 months. Hyperglycemia was associated with increased mortality. You have a 38% more likely chance of dying of pancreatic cancer if you have hyperglycemia, and faster. Association between hyperglycemia and survival in patients with newly diagnosed glioblastoma. Compared to patients with the lowest mean glucose quartile, those with the quartile 2, you had a 29% increased risk of mortality. The quartile three, a 35% chance of mortality, higher chance of mortality, and the highest quartile were a 57% more likely to pass away of progressive disease with glioblastoma. Hyperglycemia and lung cancer, a possible relationship. A possible relationship. Hyperglycemia is not uncommon in patients with lung cancer. However, it is usually overlooked. Patients with upregulated glycemia and lung cancer have been shown to have worse outcomes, reduced therapeutic effect, and more complications during treatment. This study looked at cancer as a whole. Okay, this is the relationship between diabetes mellitus and cancer and its underlying mechanisms. Epidemiological studies suggested association between diabetes mellitus and some cancers. The number of cancers appears to be increased in diabetes mellitus. On the other hand, some cancers and cancer therapies could lead to diabetes mellitus. Not good. Genetic factors, obesity, inflammation, oxidative stress, hyperglycemia, hyperinsulinemia, cancer therapies, insulin, and some other oral hypoglycemic drugs appear to play a role in the crosstalk between diabetes mellitus and cancers. If you had to dig into the paper, but it basically says that a previous large meta-analysis has estimated that diabetes is associated with a 25 to 41% increased risk of mortality or death from any cancer. So if you have hyperglycemia, if you have diabetes, you have prediabetes, you are 25 to 41% more likely to die from any cancer. Is the point being driven home at this point? Hyperglycemia and chemo resistance in breast cancer from cellular mechanisms to treatment response. At the cellular level, hyperglycemia can promote proliferation, invasion, and migration of breast cancer cells and also induce anti-apoptotic responses to enhance chemo resistance of tumors via abnormal glucose metabolism. And we see there that, again, through similar mechanisms, hyperglycemia, epithelial growth factor receptors, estrogen receptors, and insulin-like growth factors. What is this? It doesn't say. It looks like estrogen receptor again can rewire cancer to be more likely to be resistant to a variety of chemotherapies for breast cancer. Basically, you see here glucose, you see paclitaxel, you see trastuzumab, you see tamoxifen, a variety of others. And when you have high levels of glucose through all the signaling pathways we've talked about, will lead to chemotherapy resistance. HIF1 alpha expression increases preoperative concurrent chemotherapy resistance and hyperglycemic rectal cancer. Can diabetes affect the efficacy of immune checkpoint inhibitors in patients with advanced cancer? It says here that in study background, the authors explain that chronic hyperglycemia induces immune dysfunction. Furthermore, multiple studies have identified resistance or adherence to insulin therapy as barriers to achieving optimal glycemic control in patients with diabetes mellitus after frontline metformin failure. It says here that long-term, poorly controlled diabetes and hyperglycemia may impair immune checkpoint inhibitor efficacy. Strategies to improve glycemic control should be pursued in patients about to start immunotherapy. So even if you do the standard of care, chemotherapy, radiotherapy, checkpoint immune therapy, those kind of therapies are inhibited by and you're not able to get the maximum benefit from those therapies when you have hyperglycemia, when you have prediabetes, when you have diabetes. And so therefore, by putting fuel onto the fire, by being on a high sugar diet, that would only shoot yourself in the foot even more. Okay. Aerobic glycolysis. We are in the middle of a giant glycolysis series right now. And we know that the glucose transporters are critically important for cancer ability to progress. And we know that certain hormones and signaling cascades can upregulate these GLUT receptors. So here's NF-kappa B, here's PIK3 AKT, mTOR, HIF1, multiple inflammatory cytokines, as well as glucose itself. And pseudohypoxia can do it as well, can upregulate glucose transporters. And we know that overexpression of glucose transporters is associated with poor survival of most solid tumors, suggesting that expression and status of GLUT1 is a vital prognostic indicator in promising therapeutic target in solid tumors. Hexokinase is also upregulated by all of the molecular mechanisms that we've talked about that can be upregulated by any pseudohypoxic reason, but also through hyperglycemia. And we know that upregulation of hexokinase not only gives the cancer cell a protection and allows it to make energy, but it also allows it to progress and have other kind of like protective mechanisms. And we've looked at this kind of over and over again, and we see here the same pathways over and over again: MAP kinase, WNT beta catenin, AKT mTOR pathway, androgen receptor. Here's inflammatory and hypoxic and pseudohypoxic pathways. They all coalesce onto the glycolytic enzymes as well as the glutamine enzymes as well, as we've seen. And that upregulation makes cancer more robust, and it makes it more, it gives it a more of a chance to be chemo and radio and immunoresistant. Here's an association between hexokinase and tumor drug resistance. We have videos on this, so I don't really want to like dive too deeply into this. Phosphofructokinase, another key enzyme. It's important for redox homeostasis within cancer cells. PFKFB, as you can see, PFKFB is very important for the Warburg effect. It's upregulated by hypoxia, pseudohypoxia, growth factors, inflammatory cytokines, but it's also upregulated by glucose. It's a feed-forward mechanism. Hyperglycemia induces PFKFB overexpression, promotes malignant phenotype in breast cancer through MAPK activation. My point is that insulin by itself. So when you have high glucose, high insulin will also upregulate a lot of these pathways, PFKFB being one of them. Growth factors will upregulate other downstream parts of glycolysis, such as PKM2, which we're in the midst of talking about right now in our series. It will also upregulate lactate dehydrogenase, et cetera, et cetera, et cetera. And we know that the upregulation of lactate dehydrogenase is associated with poor prognosis in cancer patients. And you look at the genes or proteins or signaling cascades that are upregulating lactate dehydrogenase, the same ones over and over in HIF1, c-MYC, estrogen, epithelial growth factors, etc. So there is a true method to the madness that has been proposed by Seyfried and his group. And the foundation of that method is a calorie restricted ketogenic diet. A ketogenic diet, just for a refresher, is a low carbohydrate. In fact, it's a ultra low carbohydrate diet. It is a high fat diet. It is a moderate to low protein diet. And that ketogenic diet is the foundation. It is not the treatment of itself. It is a key component of the treatment, but is not the treatment of itself. It is the foundation of the treatment. And it takes advantage of all of the things we've talked about, with glucose and glutamine being the major substrates that are used as energy production, but also for biosynthesis, for protection against oxidative stress through glutathione, et cetera. And what ketogenic metabolic therapy is effectively trying to do is it's trying to take advantage of the Achilles heel of cancer, which is this glutamine and glucose story. And so if we are going to give you massive amounts of glucose, you're going to have a even a turbo charging of the vicious cycle that's already set up in your body. So I think that hopefully explains in gruesome detail why and how a high carbohydrate, high sugar, high processed sugar diet, if you have cancer or at risk for cancer or in remission from cancer, is a horrific idea. Okay. Which gives us to the final verdict, which I've kind of been making the verdict throughout the entire video, hopefully. But the final verdict is this is an absolutely horrific idea. Okay. Horrific idea. I know that there's a lot of influencers and people with large followings that are promoting this as the next best thing for health. And I want to believe that they are well-meaning, that they actually believe and are convicted that this is something that is really going to be helpful for you. And again, could there be a healthy version of a high carbohydrate diet? Maybe, for the right person, in the right environment, with the right kind of mitochondria. But for anybody who is at risk, high risk for cancer, is anybody who has cancer, who's in remission from cancer. I personally believe it would be probably one of the more catastrophic things you could do for yourself. And I hope that at this point, I've made that case very strong. If you like videos like this, please like, share, and subscribe. And until next time.